presentation. So Susie, speak directly into the microphone.
Okay. Well, it's really exciting to have so many people here tonight. After a couple of years of Zooming, it's taking us a while to get our audience coming back. So thank you all for coming and for finding your way into this mysterious building. So tonight we have a great program, and the title of it is Mesquite Mysteries: Form, Function, and Restoration of Riparian Bosques. Mesquites are some of the most flexible and adaptable plants on the planet, and some species can grow to the height of tall forest trees, and others just to knee-high thorny shrubs that we can run into. This flexibility has led to confusion among researchers and conflict among land managers. For example, forests of large spreading mesquites used to shelter the floodplains along terraces of our Southwest rivers. And today, most of those large trees are gone. They've either been cut for wood or plowed over, or now dying from overpumping of groundwater. And at the same time, small mesquites and shrubs have invaded millions of acres of grasslands. And their expansion into the grasslands causes concerns over watershed health and habitat. So this talk aims to make sense of mesquite variability by describing how mesquite form and function changes from the valley slopes to the river bottoms. And it'll include some tips on how to recognize these old riparian bosques. And then also we'll dive into the mesquite dynamics of a restoration site along the San Pedro River. And at this site, Nature Conservancy is partnering with the University of Arizona. And they're doing a lot of cutting edge research comparing eco-hydrology and wildlife in old growth bosque stands. And something that this talk will also cover, and some of the questions that people have had, is how you can use some of the new eco-hydrology methods to reveal secrets, like when trees are using groundwater resources and when they're using rainwater resources. And tonight our presenters are Dr. Gita Bodner, and she has been a conservation biologist with the Nature Conservancy for the past 17 years, specializing in collaboration and science-based adaptive management of arid lands. Gita got her start as a child in the woods of northern New Mexico, and she has been active with the Sky Island Apache Highlands region since 1993. Her PhD research, and I think this is really interesting to me too. Her PhD research was on jumping spiders, and it indulged her love of little things that run the planet, and also taught her to make the most of limited data in an unlimited world. Our other presenter, her co-presenter, is Collin Gillespie, and he is a master's student here at the University of Arizona, studying watershed management and eco-hydrology through the School of Natural Resources and the Environment. And Collin has worked with natural resources in different capacities for the last 10 years. He's currently researching water use patterns of mesquite along the lower San Pedro River, which Gita reminded me that the lower San Pedro River is actually the area north of Benson. It's not what we usually think of as the lower being to the south. Collin's an avid kayaker and angler, and a lover of all things water related. And yet he's chosen to move here to the desert and study arid land vegetation in a river that almost never has water in it. So please join me in welcoming Collin and Gita. Do I need to do this recording?
Already. All right, and you guys on chat or on Zoom, yeah, do let us know if the sound is going wonky besides when we move the microphone around. All right, I'm gonna just check on the chat here. Looks good. Okay. And here we go. From beginning. All right. Do you guys on Zoom see the main screen? Now I can't hear them. Will we know it if they don't? Oh, shoot. Yeah, thanks. Share screen. Of course you can't. I haven't shared the screen. Share screen. Okay. Share screen. Share. Then
That's good now.
from current slide. Okay, that's working. Yay. Okay. Okay, cool. Oh, and the chat shows up on here then otherwise. Okay, and or yeah, how do you do there? Perfect. Okay, I'm gonna pull this over here so I can see. Okay, so Susie, thanks. That was a great introduction and gave away a bunch of the secrets, but there's plenty more secrets where that came from. So what I'm going to do is I'm going to start, we are going to end up at a restoration site where Collin and I and some of our other colleagues are doing a bunch of experiments and restoration treatments right now as we speak. But to get there, I'm going to take you guys on a little journey into the onion. We're going to peel back some of the other layers of complexity that Mesquite presents to us as a way of sort of clarifying what other places and what other situations might be similar to the one that we're going to talk mostly about, and what are almost certain to be different. So I'm going to peel back the onion, and I'm going to peel it back kind of quickly so it doesn't make us cry. And Collin's going to let me know if I go too long, because there's rabbit holes everywhere you look with mesquites. There's interesting things to distract us and yeah, there's a never-ending there's no end to the places that our curiosity can take us. So it can be hard to stay on track and go quickly through stuff. So here we go. Let's see, is this changing? This is not changing try the mouse button. Okay, no, oh, come on. Next, oh, that's going to be silly. All right, so as Susie mentioned, when we talk about mesquite, we could be talking about anything from a towering tree to a knee-high shrub. So as plant people, right, our first well, okay. And when we're listening to our friends and neighbors talk about mesquites, they may be talking about them as the best things that ever existed on the planet, or absolute total villains who are ruining everything for everybody, the world's worst. So to kind of start to make sense of some of that variability in form, function, and perspective, this is a sort of a preview of the talk. I'll start by as any good botanist would do, talking about species and ecoregions and making a few distinctions. Then we'll get into where on the in a given watershed different growth forms occur. And then we'll dive into our site and to all of the activities on the site. So, all right. So what exactly is this thing we're talking about? If you search mesquite under SciNet herbarium database that most of you guys have probably spent some time at, this is what you're going to get. You will get dozens of different names that have been used for things in this region. And to make matters even more complicating, you'll see a new change from the genus Prosopis to the genus Neltuma. So you might just throw up your hands and run away screaming right then and there. Fear not, there are some patterns in this noise. It turns out that most of the native species in our region, most of the trees that are native in our region that are mesquites, are either in this glandulosa complex. You can lump it all into one, or you can divide it up into dozens of different things. But glandulosa is a thing. And so that's the blue, that's the blue polygon there. And in this nice happy red area, that's our velvet mesquite. That's Neltuma now velutina. You do get hybrids. Mesquites like to hybridize. They're very promiscuous. They'll breed with anybody in the same genus. So you get all sorts of variations caused by hybridization, especially in settings where people have planted a bunch of different stuff. But when you're out in the wilds, you will mostly see velvet mesquite, or you will see honey mesquite or some variation on it. It's worth mentioning that even within these same species, they can act very differently in different regions of their range. And so we're going to be focused here on Apache Highlands, Sky Islands area. And there are some differences between how mesquites play in the world in the Apache Highlands versus in the Sonoran Desert and Chihuahuan Desert. And I don't know nearly enough about the cinnamon and thorn scrub, so you guys can tell me about that. So, so some of these things, if you have experienced mesquite functioning differently or interacting differently, it may be that some of those observations are from different areas than this, and I would love to hear about them. So, all right. So enough with species and ecoregions. We know where we are, but where exactly are the mesquites? We've got mesquites in the uplands, up on the slopes and the bajadas. This is a semi-desert grassland transitioning into Sonoran Desert, Chihuahuan Desert. We've got mesquites that line the washes, and we've got mesquites on the river terraces. Which brings me to an attempt to sort of distinguish these different landscape settings and try to make some sense of the literature that's out there based on these landscape settings. When you go into the literature, people almost never say whether they're talking about mesquites that are in one of these different settings, if they're upland or riparian. So it's taken some work to figure out who's talking about what. So, okay, all right. So small shrubs in the uplands, bigger trees along the washes, forests of big trees on the riparian terraces. So this little diagram, I'm going to go through a few different attributes of mesquite and make some distinctions according to these three landscape settings. All right. I'll just talk about vegetation and water dynamics mostly today, but there's other ways in which these differ. And then the end of the talk is going to all be focused on the riparian stuff, the river terrace bosques. There are some ambiguous settings as well that don't fit neatly into these categories, and I would love to hear you guys. I'm sure you will have observation that you say, well, I don't know about this. So I'm eager to hear your ambiguous settings. So, all right. So to start out with vegetation, as you guys know, and as you saw in the pictures, upland mesquites tend to be small-ish. They may be small trees, they're often shrubs. And one of the interesting things about them is in semi-arid grasslands, around here, they get to a maximum cover of only about 30%. Any more than that, and there's just not enough water to go around. So if you see mesquites in stands that are much denser than that, you're either somewhere else, maybe you're in Texas where it rains more, or there's something else going on. Again, the larger trees along washes mixed with other things in linear strands. And then our great bosques would get, the trees can get to 30 feet tall, 50 feet tall even. And you can get almost continuous canopy cover, much higher canopy cover than in the uplands. And they've got their own pretty great understory. When you hear people talk about mesquite encroachment or woody plant encroachment, they're mostly talking about these uplands in the grasslands, not so much in the desert. Like Sonoran Desert, doesn't seem to have been much increase in mesquite. So when they talk about encroachment, mostly it's this setting, but occasionally others. So we're curious about that. They bring different wildlife values, and they have had different trajectories in each of these settings. In the uplands, in semi-desert grasslands at least, there's a lot more than there used to be. In the washes, we don't have much evidence of there being a lot of change in abundance, and we do recognize that they have a lot of wildlife values. And in the bosques, a lot less bosque now on the landscape than there used to be, and very high value for species that often live nowhere else. All of this is basically based on water access, and which is why we're so excited to have Collin talk about the eco-hydrology. And one important thing to recognize and to think about when you're reading anybody else's papers or when you're seeing mesquites on the landscape is that if you're a plant, unless you are near a wash or near groundwater, the only water you have access to is rainfall. And so mesquites cannot, in the uplands, cannot have evapotranspiration that's higher than precipitation. And you'll see in the literature, there's a bunch of cases where people have used the wrong evapotranspiration values because they used the values for riparian mesquites and they applied them to upland mesquites. So that's, it's a little bit nuts, like, well, wait, how could it have that much water? So, and by that same token, for the most part, in the uplands, they're not going to be affecting recharge because all the water that falls in our hot uplands, most all the water is going to either transpire through plants or it's going to evaporate. So yeah, but they do some cool things for storing soil moisture, and Collin is going to talk about that some. In washes, they can get bigger because they can drink runoff as well as the rainfall, the stuff that runs off the adjacent uplands. So evapotranspiration can be sometimes higher than precipitation. And then when you get down to the river terraces, they are going to be drinking groundwater as well as rainfall and runoff, and evapotranspiration can be much higher than the precip that falls on that acre. And when you get major declines in groundwater, they'll suffer, and we'll talk about that a little bit more in a bit. So, all right. So, how about some pictures of these guys? This is from one of our from our site that we're going to dive into in a few minutes. Big trees. How many of you guys have spent time in riparian bosques?
Where is it?
So this site is 20 miles north of Benson. And the San Pedro River has had historically some of the let's see, let me see if I can say this correctly. The San Pedro River has historically had a lot of mesquite bosques along the terraces, all along the length. Those have been cleared or lost for some other reason in a lot of places, but you will still find in a number of places some really fantastic examples of these mesquite bosques on the San Pedro. Thanks to Dale for this, for these pictures. One of the major sources of losses of bosque is just pulling the water out from underneath them. They need to have access to that groundwater, or they won't thrive, or at a certain level they won't survive anymore. So ideally, 15, 20 feet. They can do okay at 50 feet. They like it closer to the ground than that. Clearing for agriculture has also been a major source of loss of mesquite bosques because they're on these nice, flat, very fertile terraces. This is at the north end of the lower San Pedro up towards Mammoth, which brings us then to our site itself. So Tucson, you can see yourself in Tucson right over here. You can see Wilcox Playa. This green band is the San Pedro River, and our site is right here in the lower San Pedro, where, let's see, okay, there we go. So this is a property that Nature Conservancy bought in 2002, partly because it had good shallow groundwater, and partly because that water was being actively pumped out from underneath it to irrigate 300 acres of farm fields. So at that time, we were working hard to try and figure out how to keep more water in the river. And Nature Conservancy and a bunch of other groups got together and tried to figure out, well, how can we reduce the amount of that water that's pulled directly out of the river subflow and used to water cotton and alfalfa and things like that. So when TNC bought the property, we turned off the water and stopped the, mothballed the plows, sold the plows, I suspect we didn't even buy the plows, and waited to see what happened. And we're gonna, there we go. A little orientation. This side of the river, the west side of the river, has old growth bosque on it. The east side of the river is this 300 acres of old farm fields that have now, as you can see from the aerial, grown up in also mesquite, but rather different than the bosque on the other side. It was also bosque before it was cleared for farming, according to the folks who've lived there forever. So it was farmed from about 1950 to 2002. So just a couple pictures of what this looks like before I turn it over to Collin to do his portion. This is just a glimpse of what the old growth looks like. Big, shaggy, spreading trees with fantastic understory. There's a broad diversity of really cool shrubs in the understory. There's all sorts of vines in the understory. And I figured a botany group would appreciate this the way that nobody else appreciates this. The sticker fields under there, in the understory of the bosques are sometimes pretty gnarly. Anybody want to identify the plants in there based on the stickers? Mm-hmm, yep. Yep. Mostly those two. They are extremely prickly. This is this is one bosque plot sort of seen through time. You can see that that bosque has been there. So our earliest really good pictures are from aerial photos are from 1996. It has not changed a bit from the sky, at least in those 26 years. And I suspect it has been pretty stable for a lot longer than that. In contrast, this is what's happening on those old farm fields. We've got some areas with very dense young thickets. All of the trees in those thickets in those farm fields are 20 years old or younger. And it turns out most of them are almost exactly 20 years. They came in really quickly, and it can be an impenetrable mess. And so one of the goals of this project was to figure out, as obnoxious as it is for us humans, what value does it have for wildlife? We were really curious to see, what good are these? And we've been asked many times by neighbors, what good are those thickets? So we wanted to try and answer that question. Understory there is a glorious combination of amaranth, so pigweed, Russian thistle, a little bit of Bermuda grass, a bunch of other annual grasses, all the stuff you want to roll around in. But there's some really great understory in the second growth thickets as well. Some sharp-toothed understory as well. So. All right, and looking at the same time period on these guys in the second growth, you can see tremendous and very rapid changes. From 96 when it was actively farmed, 2002, this was probably right around when we stopped farming it. And you can see that those trees come in pretty quick, and then there's some crazy things like the same number of trees pretty much, I would guess, in 2019 from 2017, but they're all much bigger. And then 2022, this was from right after we finished a thinning project, a little thinning experiment that Collin can tell you more about. So yeah, so across this site, we've got a lot of riparian mesquite with a really big range of ages and densities, and felt like it was a perfect place to try and understand what riparian mesquite does as it gets older. And yeah, and with that, I'm going to turn it over to Collin. Any questions for me while he's? Yeah.
Did those trees just come in on that farmland spontaneously? Or
They-
did you plant it?
They came in all by themselves, and probably with the help of some cows. Yep, we did not plant anything out there. So it's kind of amazing. I mean, there's plenty of old farm fields that would still be just dust and weeds. And so to have it come in that thick with native vegetation of any type is an accomplishment.
Yeah, and so densely at
Yeah.
that.
Yeah.
Any other questions? Yes, sir.
I have a question from chat. Jan
Yeah.
Turner is asking, how did you turn off the water at the TNC property?
Well, Dale might have to help me answer that. Were you there at the time, Dale?
Turned off the pumps.
Yep.
It was easy.
And tried to work to get the water rights transferred to the river, but I think that the paper side of it is still in process. The goal was to leave the water for the river rather than taking it out for alfalfa or whatnot. But yeah, pretty much just turned off the pumps and stopped plowing, and all this crazy stuff happened since. Yeah.
Is there any record of some sort of acid bed or mesquite somewhere like dying for some reason that end up doing like not the mechanical or chemical removal, but some disease or anything that affects these trees?
I've not heard of any major disease die-offs. You definitely see places where they've died or died back, like a lot of branch dieback when the water table's gone down and when there's been big droughts. You can see that on Cienega Creek Natural Preserve, for example, even from I-10. You can look at them and you'll say, well, those are big trees and the tops are all bare. So that's the lowering of the water table and drought on that. And you can see some places where they've died entirely from drought and lowered water table. There would have to be a lot of fungi, I think, that would affect them, but they're awfully tough. Their wood is so hard. You can tell they put a lot of work into making themselves tough. Yeah.
People on Zoom are not hearing questions from the audience, so if there's a question.
Okay, we'll repeat the question. Okay, yeah, that question was about are there things that kill mesquite besides us intentionally trying to kill mesquite? And the question before was about whether we planted any of that or if it just came in. All right.
Thank you. Sure. Thank you. Okay, so Gita mentioned that sometimes mesquite are portrayed as a little bit villainous at times. There's a suspicion that they can be limiting the amount of water we see in streams and rivers. And so there have been projects throughout the Southwest, sometimes on the state level or the private level, to remove mesquite for the purpose of like restoring more stream flow. But it's really not established in the literature that it would have this effect. And so it's kind of in that context that I'm doing my master's project, looking to see, at least trying to answer a piece of that question. Do mesquites use a source water that would otherwise become stream flow? And so that's what I've been working on since last fall. And we're looking at different sources of mesquite to see if that's something that would justify their removal or if it would make much more sense to try to preserve them. And so I'm going to talk a little bit about our study, some of the measurement techniques that we use, and some of our preliminary findings. So we're investigating the source water. Hold on, where am I at? There we go. We're investigating the source water patterns of mesquite to see if or how they vary from young trees to more mature trees, how they vary throughout the year, and if they vary throughout the year, if a thinning treatment would impact the water use patterns of the younger trees and how water access impacts tree water stress. So, and by investigating source water, I mean, where are the trees getting their water from in the soil? Are they getting it from shallow layers? Or are they getting it from deeper in the soil? And then does that pattern change throughout the year? Oh, and those are also some of the questions that we're looking at. So we're studying 4 distinct stands of mesquite at 3 links that's located along the San Pedro River, and Gita kind of mentioned all of them. Let me see if I can use this. So just like she said, on the west side of the river, there's this mature bosque. And the ones in the plots labeled with stars are the ones that we're actually testing in specifically. Then along the east side, this is what was the cultivated field. It's now grown back in with some thicket, and especially the southeastern corner is especially dense. Down here is where we have our control. That's just a dense thicket. This here is a thinned thicket where we've removed, Gita, how many do you think? Like a quarter of the trees, a third, something like that, to see what impact that would have on the growth of the others. And then we're also incorporating a look at an upland plot to give us a little bit of comparison like that. Let's see this. Okay. So you already saw some pictures of what some of the different stands look like, but I wanted to show this one as well because I feel like it does a pretty good job. So the mature bosque is naturally thinner and it contains larger, older trees as you might expect. It's got a fairly thick canopy and a diverse understory. I like working in that one the best because I don't have to stoop as much as I do in the other ones. The dense thicket is, like you'd expect, really dense. It's very difficult to walk through. The trees are mostly smaller. It's a really dense canopy, even denser than in the mature bosque. And there's not much understory in the dense one. And then the thinned young thicket is, this one was naturally a little bit thinner anyway, but it has undergone a thinning. We removed a bunch of trees last spring, and now it has a much more diverse understory than the dense thicket does, and a much more open canopy. And so last May, we started collecting meteorological data at each of our test plots, including measurements of air temperature, relative humidity, soil temperature, soil moisture, and other things besides that, all to get a better look at how the trees are going to be responding to varying meteorological conditions at each of the plots, and potentially how the different stand structure might impact some of those conditions, like moisture, like temperature, stuff like that. And so those are done with these like automatic data loggers that are connected to sensors buried in the ground or on the post. And they're logged every hour. And I go out about every 6 weeks to collect that data. So some other things that we're measuring. We've been measuring a leaf area index to get a little more quantitative comparison of the stand structure to see how those canopies vary. Some are more open than others. And for that, we're using this LICOR device, which measures the light coming through the canopy to give a number of the actual density of the canopy. We've also been using dendrometers. These are used to track the growth of the trunks of some of the trees in our test plot. These devices have a metal band that you can see right up here that wraps around the trunk, and it connects to this piece in the middle that has these measurements. And so as the tree grows, it's able to slide apart to track the change in growth, and actually can slide back as well for whenever the trunk contracts a little bit. And so yeah, we installed those last March, and we were out there in October and could see that this one, for example, we set them at 5. So this is where we started them. And you can see it's slid over a little bit over here to 6. So there's a little bit of growth on that one. And then I saw another one as well that looked like it has grown substantially, but I'm going to try to look at them a lot more closely next time I go out there and actually record those values. I think, oh yeah, my battery's dead, but I can try to project and hopefully everybody can hear me online. It's okay. I was in theater in high school, so I trained for this. So just feel at ease. Oh yes, so like with many other techniques, mesquite are not always very wieldy for some of the measurement techniques we want to use, and that certainly applies to dendrometers. You need a trunk, a single trunk, ideally at chest height, for these dendrometers, but that's not often easy to come by because mesquite often fork below or right at ground level. So you can see in this picture, you know, we found some that works on, but it's not always easy. But we thought it'd be worth trying because we wanted to see if perhaps some of the trees that exhibit the most growth might use a specific water source, but that might be interesting. Oh, and in fact, that reminds me, if we've got time, Gita wanted to put out a call for help. We want to maybe institute some DIY dendrometers that maybe you guys might be interested in helping us with. Another measurement technique that I think is really fun and interesting, it's very, to me, very, very stereotypically sciencey, is measuring leaf water potential using a Scholander pressure chamber, this thing right here. This technique is used to get an indication of a tree's water stress. It's done by taking a cutting, like you can see in this picture here, and you seal that cutting inside this chamber, and there's a gasket that seals around the cut end of the cutting. And so you place it down in that chamber, and we fill the chamber inside with nitrogen gas and slowly increase the pressure. Thank you very much. And you slowly increase the pressure, and the gauge reads how much pressure there is, and the amount of pressure it takes to force the moisture out of the cut end of the stem is a measure of how much tension the tree requires to move water through its tissue under normal processes like transpiration. And so that gives us a sort of a measurement of the amount of stress the tree's under. It's not like all levels are stressful, but the more negative these values, the more stress the tree's under. What's that? Like blood pressure. Yeah, exactly, like blood pressure. And so we do this before dawn to get a look at what that stress, what that tension looks like when the trees are at their least stressed of the day. And we do it again at midday to see what they look like at their most stressed. And I have to say that waking up at 3:30 in the morning to run around in the dark, measuring these trees has not been my favorite part of the study. It's more like type 2 fun. It's maybe fun later, but while happening, not that fun. But it's a nice look at the bosque, a different time of day. So I was going to show you a look at what the water potential data looked like, and then I thought that was a terrible mistake, and you wouldn't want to see that because it looked super not exciting. But let me just tell you in brief that I've already been surprised to see that the more mature trees seem to experience more water stress than the younger trees, which I did not expect. I would expect that the younger trees being able to root, that the more mature trees being able to root deeper would be much less stressed at all times of the year, is what I expected. But that has not been the case so far. And as the monsoon season went on, all of the trees seemed to become more stressed, which again was a surprise to me. This could indicate that the more mature trees and the others as well are not taking advantage of the monsoon precipitation. But we're hoping to be able to confirm this with our stable isotope analysis, which I'm going to talk a little bit more about. It could also suggest that the mature trees especially do not need to switch their water source as the monsoon goes on, despite appearing to be more stressed, perhaps because they can tolerate much higher levels of stress. They are, after all, a really arid adapted species. Okay, so now I want to talk about, this is kind of the real crux of my project, and consequently, or not consequently, but incidentally, more of the boring part. So please bear with me. I want to try to break it down. And this will be good practice for me explaining to people who may not otherwise know. So I mentioned the isotope analysis. So I want to explain a little bit about how we use it, because I wouldn't expect you to know, as I certainly did not know when I began my project. Basically, isotopes are variations of chemical elements that have the same number of protons, but different numbers of neutrons. So in this image, you can see I've got some pictures of isotopes of oxygen and of hydrogen. So oxygen 16 has 8 electrons, 8 protons, 8 neutrons. Oxygen 18, 8 electrons, 8 protons, 10 neutrons. And these stable isotopes that we use, they're naturally occurring, and you find them all the time. In a given drop of water, some molecules are going to have these 8 neutrons. Some of them are going to have 10 neutrons. And so that combination that you see in different water, we would call that like an isotopic signature. And water in different places has different isotopic signatures. Groundwater, that's going to look differently than, say, precipitation or water that you would pull out of the atmosphere in a cloud. Each of those has a combination of these oxygen isotopes and these hydrogen isotopes that we can think of as a signature. And so using that signature, we can draw some conclusions about what's going on with the trees. By taking samples from the trees and extracting the water, we can compare the isotopic signatures of the water within the trees to the samples that we take of precipitation out at 3 links, samples we take from the groundwater, from the wells that are on the property, and from shallow soil pits that I dig at each of the plots. On many species of trees, this wood sample can be taken by coring, but we found coring to be very difficult on mesquites. They're very dense. So I use cuttings from the branches and extract the water in my lab. As you can imagine, actually, as I mentioned, that large trees can root deeper than small trees, but that does not necessarily mean that they will draw all their water from deeper sources. And so we're tracking those use patterns to get an idea of where that water is coming from. Okay, it's gonna get a little worse before it gets better. Now, I want to look at a little of my isotope data, but first, I'm going to try my best to walk us through a short primer on how to read this dual isotope plot. Don't worry about the axes. It's not very important for us. Just know that this line right here is called the global meteoric water line, and it basically contains values of precipitation from across the world. Colder regions are going to be down here on the line. Warmer regions are going to be up here on the line. Similarly, summer rains are going to be further up the line, and winter rains are going to be further down the line. When water is given time to evaporate, the ratio, or that signature that I mentioned, it's going to change. And so that water will drift away from the line, and shallow soil water, especially in warmer temperatures, is going to give us values out here in this direction. Colder regions, but deeper water and winter precipitation, it's going to have values a little further down here, down and further closer to the line. So that looks, I know it's a little bit squirrely, but so now I want to show you that so I can show you what some of my data from 3 links looks like. And I realize it's a little busy. There's a lot going on here, but let me briefly point out just a few interesting trends that we may be seeing so far. So these figures have the same layout as that big one on the last slide. And keep in mind that the dots represent the water taken from the trees, and the asterisks represent water from the soil, the shallow soil. And then this plot down here labeled rain, those are the actual rain values. And so just to point out a couple things, both in January and March, so by the way, it's color-coded by date. So January would be kind of this, does it? Oh yeah, it's not quite as good on the screen. I anticipated that, but didn't do anything about it. So this looks like January right here. It's kind of a salmon color. And this over here is March. So as the water taken from the trees is for the most part, in January and March, it's a little further away from the line, indicating that they're using older shallow soil moisture that has had more time to evaporate. And they're relying a little bit less on the recent rain. The exception you can see is this mark down here, that it's very close to this value of precipitation for January. So that would suggest that that tree is using that water very rapidly, which is not the same as the other trees from that plot. So I looked and found that that was one of the smaller trees that's in the mature bosque. It's one of the smaller trees, and it's actually a really sickly tree that I'm really worried about. I'm afraid it's going to die on me and not make it to the end of my project. But so that left me wondering, why is that? You know, maybe there's a reason why it's using more of that shallow water. And I don't know what that is yet, but it's just something that I plan to keep in mind. Let's see, by April, which is this olive color out here, here, some of these here, over here. By April, values start moving back in the direction toward the line, which indicates an increasing reliance on the deeper soil moisture, which makes sense because those shallower soils are probably starting to dry down. April's warming up. There was not a lot of rain out there during that time. Also in April, the trees in our upland site, they're really split. There's a couple that are drawing water from way out here, a couple from way over here. Their size does not vary that much. So as I get more data, I'm going to be looking really closely and seeing why that might be. That it seems like a greater discrepancy there with that plot in April than in some of my other ones. And when the monsoon rains of July come, which would be this value here, If the trees were relying on that monsoon rain in July, then you would see more of their points in that region. I know you can't see the color well, but there's a few of these that are from July that you would expect to be over there if they were using that, but it appears that they're not. And so that was a bit of a surprise to me. Now, I've only just begun to analyze this data, and I have a lot more to do that requires a lot of careful interpretation. This technique is really powerful, but it is not, it certainly has its limits and what it can tell us. And some of the trends are obviously not as clear as I would prefer them, but that's part of it. There's a lot of confounding factors, such as the time between a precipitation event and when I take my samples. The longer that time period, the more the values are going to diverge. And so it makes the trend a little more difficult to see. We also need to see if maybe tight groups of points represent the same few trees, or maybe if tree size has an impact on where they're drawing their water. And we'll need to look at more closely at the differences in soil composition, because I know the soils at the Bosque plot are sandier than the other plots. So that might be something that's going to have an impact as well. The last thing I want to talk about is another potentially confounding factor that we need to take into account, and it's called hydraulic redistribution. And I want to describe it briefly because I think it's really fascinating, and I would expect you would as well. Hydraulic redistribution refers to the upward, downward, and lateral movement of water through plant tissue, specifically the roots. And it's distinct from the movement that occurs during transpiration because it happens while the stomata in the leaves, or needles, as this species may have, are closed. And it even can occur when trees are dormant during the non-growing seasons, which I think is pretty interesting. So this movement occurs, actually, let me point this out in the figure real fast. So in the dry season, you can see that at night, this water is still moving up the roots, just like it would during the day. But instead of going up into the tree, it's being pulled out into these shallower soils, presumably because in this situation, it's more moist down here in these deeper soils and drier up here. So that natural difference in the soil moisture gradient is pulling that water through the roots. And the wet season would be the opposite. Maybe the soils are drier down below and wetter on top. And so that same process is pulling water from these shallower layers down into these deeper layers. And we could say that the trees, whether intentionally or not, are banking that water for when the dry season comes back around. This movement occurs just because of a natural soil gradient. This process can be both competitive or facilitative for understory plants. It can help bring moisture up to them whenever they can't otherwise reach it. But then there's other times, like say in the wet season, where they're drawing that water away. So the whole reason I bring this up, though, is because this can confound some of our data. Because as you can imagine, if a tree is pulling water from shallow soil moisture layers, but there's some of this redistribution happening, and water from deeper in the soil is being redistributed to those shallow layers, it's going to throw off that signature, and it's going to make it less clear than it would have otherwise been. And so it won't be as obvious whenever we're making that comparison. Is that just shallow soil moisture, or is it mixed with deeper moisture as well? So basically that sums up my part of the project focused on ecohydrology. It's still very much a work in progress, and I'm looking forward to processing and analyzing more of my data in the coming months. And maybe you could have me again next year and I could talk about what I found by the end. But now I'm going to pass it back to Gita. If we've still got, we basically don't really have much time, but to talk about any of the other aspects of the project or anything else.
Now that the music behind us is really ramped up. All right, so we figured that we might use all of our time on this part, but we stashed a whole bunch of slides on other aspects of the project at the end. So I'm gonna, there we go. I'm gonna put up the acknowledgement slides, and thanks for all the folks that we've been working with on this. And then if any of you guys wanna stay a little longer, wanna find out what we're finding about the birds and how the birds differ from thickets to old growth bosque, or want to hear about how the birds are responding to the thinning, or want to see how well mesquites also hide their secrets when you look at tree rings, or want to know anything else about vegetation characteristics in the different plots, we'd be happy to talk some more on those. So, yeah. Okay, yeah, let's do some questions. Yeah, let's do some questions. And one of them could be like, show me the slide on such and such.
I didn't see any reference to biomass in your studies.
Yeah,
And I was wondering if you saw any trends. Because I'm interested in getting the carbon out of the atmosphere.
yeah.
So how, have you seen anything you can talk about on that?
That's a fantastic question. The short answer of that is that we are still trying to figure out how we're going to estimate biomass. There have been okay. Oh, sorry. The question was about biomass. What's the biomass doing out here? Are we getting massive amounts of carbon capture? Can we say anything else about how this system might participate in pulling carbon out of the atmosphere and saving us from ourselves? The short answer is there's a lot of biomass out there, but we haven't yet figured out exactly how we're going to estimate that. There are equations that have been developed for upland mesquites, and so we'll be able to make some estimates based on those equations. There are ways of translating the canopy dimensions and diameter at breast height into biomass estimates, but we're pretty sure that they're not going to be quite right for these riparian trees. And we have some folks who specialize in this who are going to come down from ASU and help us try and figure that out. And if anybody else wants to help us try and figure out the biomass side of it and how to estimate it in a system that we can't assume is like every other system, that'd be great. And we also want to we've started taking some soil samples to look at soil carbon, because there's been great work out there that shows that mesquite can be very good at stashing carbon in the soil. And so we would like very much to see how our different stand types compare in terms of soil carbon, and how what the pattern looks like for that accrual of soil carbon. All we've done so far is take a handful of samples and then beg some experts to come and help us figure out how to do it better. Yeah.
We've got two related questions from online audience
Okay.
members related to fungi. The first question is, do fungi play a role in mesquite biology? And then the second question is, how do mycorrhizal networks factor into these mesquite bosques? In many areas, the fungi broker exchange of water and nutrients underground.
Well, I would love to know the answers to those too. Okay, questions being, what role do fungi play in these mesquite bosques, and what role do mycorrhizal networks play in the network of trees and their interconnectedness below ground? I have no idea. I'm sure there are a lot of fungi, and I would love to know some of the answers to that. These guys they're nitrogen fixers. Let's see, what's somebody refresh my memory. Who does the fixing of the nitrogen on their roots? Yeah. Probably mostly bacterial rather than fungal nitrogen fixation, right? Yeah. And I have no idea on the rest of the fungi question, but I'll bet there's a lot, and I'll bet they're different in the old growth to the young growth. And if anybody wants to come and take some samples and try and help figure that out, we've got the perfect site for you.
And we've got a follow-on question from an online audience member.
Yeah.
Where are the nitrogen fixing bacteria? Are they shallow or deep? And if you could repeat the question.
Okay, where are the nitrogen fixing bacteria on the roots of the mesquite? Are they shallow or are they deep? It has never actually occurred to me to wonder which roots they're on, which is kind of, yeah, anybody here, anybody else in the audience know? And I would assume that they are at least in the shallow roots, on the shallow roots. Yeah, you guys may have figured out from this, and you may have already known, that mesquite have both extensive lateral root systems and deep tap roots. So yeah, is one of those sets of roots more active in nitrogen fixation than the other? I have no idea. Anybody else know? That's a great idea. Great question. Yeah.
I'll ask a question you can't answer.
Okay.
What about those wildlife values of those dense thickets? You know,
Wildlife values of the
What-
dense thickets. Yeah, cool. Let me get you to the bird part. Yeah, so we have a bunch of wildlife cameras out now, and we have yet to go through all of the photos for them. So hopefully in a few months, we'll be able to tell you more about other wildlife. But the bird studies, so we've had breeding bird surveys out for 2 years now and just got the data crunched on those. And the short answer is that both the mature bosques and the second growth thickets are excellent bird habitat. That there's approximately the same diversity of birds in both the old growth and especially the dense and moderately dense thickets. But there's some differences in there. So the bosques overall have higher abundances than the thickets, and there are certain birds that have preferences. So this is work that Aaron Flesch just wrote up, that of the 28 most common breeding birds out there, 10 of them seem to be actually don't particularly care whether it's a bosque or a thicket, old or young stand. 12 species are more abundant in the older stands. 3 species are more abundant in the densest thickets. One of them really likes the middle ground, like the Goldilocks. And then there are some that you would expect, as you would expect, there are some that really only occur where there's still a fair amount of open ground and low growing vegetation. So yeah, they are, even the second growth thickets are full of birds. We can't get into them very easily. It's a pain in everywhere a spine can get you to do the surveys in the thickets, but the birds like them. So yeah, that's the short answer. We're pretty excited about it. Oh, and we also have some results on the responses to thinning. And let's see if I can, the summer tanagers and the yellow-breasted chats, and one of the other guys, Bell's vireos declined after as a result of thinning. Yellow-breasted chats, summer tanagers, and somebody else increased. Vermilion flycatcher increased after thinning. So, and Lucy's warblers. So Lucy's warblers are kind of the quintessential mesquite bosque bird. It is where it's where they live. And there are absolutely gobs of them in the old growth. There were an average of 17 Lucy's warblers in every 6-acre plot in the old growth. There's about half that in the young stuff, which is actually more than we expected. So yeah, they're pretty good. They're not great, but they're getting there. Yeah.
When you turned off the water in 2002.
Yeah.
And miraculously all these mesquites came back over the next 20 years. Didn't you get any desert broom or salt cedar coming in?
No salt cedar at that location. There are some other properties that we manage that have had salt cedar come in alongside of mesquite, and it can use very similar habitat. For whatever reason, it didn't come in here. And there is some salt cedar along the river channel, basically in between some of our plots, but none out on the flats. We don't know why.
Because in other areas we've seen it, desert broom can become very weedy. You know, in these open spaces where they can go in and colonize. Yeah,
very interesting. I think of some individual desert broom out there, but there's not large patches of it. Yeah. Some of the critters might like it if there were.
How about the other desert bushes? You know, your catclaw acacia, your whitethorn acacia.
Yeah,
Are
we
there
have
any
some,
coming in?
we have some giant old growth cat claw in the bosque, but I haven't seen any cat claw in the young stuff. Yeah, it's a little bit of one of many mysteries why we are getting certain things coming in and not others. Maybe it's seed source.
What about like a hackberry, like desert hackberry or canyon?
Yeah,
hackberry?
Yep. So in the mature bosque, there are big and sometimes small. There's a scattering of the tree hackberry, I have not seen any of those yet in the young stuff. But we are optimistic that maybe once we're sort of imagining a bit of a successional pathway once the berry eating birds. I want to talk with Aaron about which of these birds that he's seeing in both sides are berry eaters, and if they might be able to be encouraged to bring more seeds in from just across the river and get more of these berry bushes, native berry bushes started in the second growth. There is a scattering of wolfberry lycium that has started to come in the second growth, but it is that understory. Those understory shrubs are not nearly as diverse yet than in the old growth.
Desert hackberry coming in now.
I'm trying to think. I don't think I've seen the desert hackberry there either. Yeah,
Well, we do have that in the mature side, right?
we have the tree hackberry,
Oh,
the
okay.
netley hackberry. Yeah, I haven't seen the desert hackberry out there, but
wondering
now
if
I'm
it
gonna
could
look.
be that we're
Uh-huh.
not seeing the same diversity in this new like thicket area because of the same flood regimes that used to be in the river area aren't really there anymore because the flows changed. So maybe the seed dispersal just can't work the way that it once did.
Yeah, we're very curious about that.
We have a question from an online
Okay.
audience member about mistletoe. How does it vary between these different habitats?
Yeah, I have not seen much mistletoe in the second growth. Have you? And I'm trying to think, Dale, can you think of much mistletoe in the old growth over there? I'm sure it's there,
I'm
but
sure it's there, but I haven't seen it.
yeah.
But it seems like mistletoe prefers more mature trees than little ones, for whatever reasons.
Mhm.
The most I've noticed was in the upland.
Oh,
I've seen
okay.
the most.
Yeah, the uplands there, we actually had a little bit of a hard time finding plots of upland mesquite to compare with the riparian because it's far enough into the sort of Sonoran Desert transition that that most of the uplands have the whitethorn acacia, the catclaw acacia, and the Laria, the creosote bush. But the mesquites that are in the upland, in the spots where we did find them, they're pretty old. And they again are very stable. I have a similar slide of the changes through time. And with the upland plots, they look the same now as they did in 1996. So and we're eager to get some older pictures to see too, but those ones are kind of stable. So yeah, that was a question, an answer to a question nobody asked, but yeah.
Have you seen native peplums?
I don't think that they certainly aren't abundant out there. And I'll have to look at the bird list and see what we got. Yeah.
They are obligates of mistletoe. so
Yeah.
Maybe since you don't have mistletoe.
Maybe there aren't very many. Yeah. Since we don't have a lot of
but
mistletoe,
they
maybe
would
there aren't very
plant
many.
mistletoe
For sure. Yeah.
and stuff.
Yeah.
It would help
Bring
to
'em in.
spread it.
Yeah. I'm sure we've got some, but I haven't noticed a lot out there, which is kind of, yeah, makes you scratch your head, doesn't it? Yeah. Yeah.
What was the research you did on the tree rate?
Oh, cool. Okay. So let's see. Oops. Oh no, I think it's over here still. There we go. Oh, I've got a couple different tree ring ones. Let me see. Let me scroll through here and figure out which ones. Ah, yes, there. I'm going to pull one of these up to there so that I can also show it. From current slide. Okay. So while we had a bunch of guys out there with chainsaws, we politely asked them if they could cut us a bunch of tree cookies, and they did. So from each of our thinned plots, we have about a dozen slices of trees of different sizes, and we've Collin and our coworker Jesse, who's a dendroecologist, polished them up and analyzed a subset of them. So about 3 slices per plot, and found some pretty interesting stuff. Yes, you can see rings. Yay! Yes, you can count how old the tree is. I couldn't, I would get it wrong, but Jesse, who has a lot of experience in it, and was able to read the one master's thesis that's been done on mesquite tree rings, was able to identify annual rings in it. And basically she was confirmed that none of the trees that we have looked at samples for are older than 20 years, which is good because we know from that they couldn't be, right? So and that they're actually pretty tightly there also aren't very many that are a lot younger than that. So it does seem her tree ring counts supported the idea that most of the trees came in at this pretty close to the same time. Within a few years, most of the trees that we looked at least were already out there. Some of them just grew a lot faster than others. She also found that she was not able to cross date them. So if you look at the pattern of wide and narrow rings in normal trees, you can find out a lot about the climate patterns, right? You can tell a dry year from a wet year, and every tree in a stand and every tree, you know, in that region is going to show some of the same like patterns of wet to dry. And you can say, oh, well, that was 1993 and that was, you know, 2011. And you can cross date them and compare them. These guys are not yet behaving in any consistent way. You can count them, but you can't compare them across trees. So mesquites are still hiding their mysteries in the tree ring. There was a master's this is a figure out of one of the one master's thesis that was done on mesquite tree rings. And basically, I think that the poor fellow may have thrown up his hands and switched fields because they were so hard to work with. Yeah, he came to some similar conclusions. It's very hard to cross date them. His also suggested that that the cross dating got a little bit better after the trees were older than about 40 years. So we either need to get some slices from the old growth bosque and the old growth upland to look and see if there's maybe some more consistent patterns in the older trees, or somebody's going to have to invent a better way of coring mesquite trees that doesn't break off the bits and make your all of your dendrochronologists mad at you for ruining their equipment. Yeah. This also has all sorts of cool things, like he did find some neat things in here though that match with some of what Collin was saying, that we're not necessarily seeing evidence of them using recent monsoon rainfall. This graph up on the top shows correlations between ring width and precipitation, and the only really strong signal there is that the trees are putting on wider rings. To the extent that you can read anything into it, they're putting on wider rings in years after wet fall, like wet September, October, November, which is also then really a head scratcher. They're not putting on wide rings when it rains a lot. They're putting on wide rings a while after it has rained in the fall, and that's the only sort of clear signal coming out of it. Yeah.
How do you know when that it got rain there? You know, I'm just thinking about, you know, how over here it's raining and here it's not. How do you know it got rain after? Like,
We have an automated rain gauge. It's like a little automatic tipping bucket thing that collects it. And then when it fills up, it empties itself. So it's able to track individual rain events all the time.
That you said were wider in certain times.
So at our site, we would be comparing rings with the rainfall data that we've collected on site, which may not be perfect from one, from one plot to the next. It might rain on one plot and not rain on the next plot. Yeah, quarter mile away. But this study was done at the Santa Rita Experimental Range, where they've got a really dense set of rain gauges, and they know every drop that falls within, I don't know, they've got them every mile or so, I think, even. So,
But
yeah.
the trees don't go back very far. The trees are not very old.
Ours that we're looking at are only those 20-year-old trees. We only took the sample so far from the ones that we were cutting down anyway. So we haven't been able to talk ourselves into cutting down any of the old stuff. But if anybody else is cutting down one, we want to see it. If the power company comes out and tries to cut one and has to cut one down, we want to be right there behind them and get a cookie from it. Did that answer it?
Oh,
Had we- yeah. Yeah.
Any other questions? Do we have any more online?
That's it.
Dale?
Yeah, so speaking of biomass, I know it's early, but any noticeable response to the thinning in the thin plots versus the unthinned plots? What the trees are doing because of that?
We haven't yet collected no answer yet. Everybody grew a bunch last year, and we haven't gone out yet and taken the full set of measurements on the thinned and unthinned yet. I think we're losing battery on this thing again, or else I don't know how to speak into it. Yeah, stay tuned. Come help us measure.
Okay, well, thank you so much, Gita and Collin. I really appreciate your great presentation. Lots of information going on here. So,
Thank you all for the great questions.
yeah, and they'll be around maybe a little bit afterward if you have other questions. So I'm going to stop screen sharing and say to everybody out there.
There's one more question that just came up on the chat.
Okay. One more question. Gita?
Oh,
What was it?
yeah, I was under the impression that mesquite trees can have very deep roots. Are you sure that they are not getting groundwater, which would make seasonal patterns blur? We think they are getting groundwater, but we don't know when they started getting groundwater. And if the older ones get more of their water from the ground than the younger ones. So we're trying to figure that out when they start, like, and we're going to be looking at some historic imagery to also try and figure out if we can tell when the young ones started getting groundwater. We haven't done that part yet. Did that answer it, Jillian?
To some degree.
Yeah. Yeah, we're counting on them having very deep roots. We assume that at least some of them are drinking groundwater, but we want to know more than just that.
Yeah, if somebody could get some samples of that groundwater and compare the isotopic signature to individual trees, you might be able to tell how much they're getting in regard to groundwater versus surface water.
Yep, absolutely. Collin is nodding his head. We've gotten samples from the wells, and I don't know if you've analyzed them yet, but that is part of the plan. So you're brilliant, Jillian.
Yeah, I live in an area that I guess you would call whatever you call it, highland. You know, it's got creosotes and palo verdes and mesquites, and there's a lot of good sized mesquites in my area, and I think that they're tapping groundwater because the groundwater table is only like 100 and less than 200 feet deep. So their roots are getting there. And during that terrible drought, when we had prickly pears crapping out all over the place, the mesquites did fine. And I think that they had the groundwater, that's why.
That would be fascinating. If they are able to get groundwater that deep, that would be kind of amazing. And it's, one of these crazy ambiguous settings. Is it upland? Is it riparian? Are they drinking groundwater or not? We're hoping to have some better techniques for figuring that out. But we're still working on it.
Just as a, some backup to that, background to that, we keep a rain gauge at our house. And during 2020 and the first half of 2021, during an 18-month period, we did not have, we had a total of 2 inches of rain total in 18 months. Prickly pears, yuccas, Desilerion were dying all over the place, but the mesquites did great. So I really think that the mesquites are getting the groundwater.
Yeah, be great to know. They are tough as nails. I'm always impressed at what they can do. And every time you say that they can't do something, they prove you wrong.
Yeah. Thanks.
Okay, thank you for the questions from our Zoom audience as well as our local audience. And I'm going to give the microphone over to Susie just briefly so she can tell you about what's coming in December.
Thank you. So our December speaker will be Jim Verrier, who a lot of you I'm sure know from Desert Survivors. And his topic will be the distribution and drivers of subtropical plants in the Sky Islands. So these subtropical plants that come up and just make it into our Sky Islands, a lot of them are some of our favorite plants. And he'll be talking about his research in how they get here and why they're here. So we hope that, and that's going to be on December the 9th. That's the second Thursday. So we hope that we'll see some of you then. All of you then.
Okay, great. So I'm going to stop the recording and I'm going to